Okay, in this video, I'm going to be talking about this this marker called Scara5 or Scara5, and it's involved in a cancer tunneling nanotubes. So basically, it's a ferritin receptor responsible for uptaking ferritin-bound iron into cells like macrophages or kidney cells. So the normal function of Scara5 is endocytosis, ferritin delivered to lysosomes, leading to ferritinolysis. The lysosomes is they're responsible for the process of iron. There can be some issues with iron trapped in macrophages that could instead turn lysosomes into iron reactors, because they're storing iron, and they're not supposed to be doing that. That's not their function. But I'll explain in this video and future videos as well the mechanisms of iron trapped in macrophages and how that leads to lysosomes doing what they're not supposed to with the iron. Scara5 is low or absent. Ferritin uptake drops. Ferritin accumulates extracellularly, and then intracellular iron delivery is impaired, leading to functional iron deficiency in the cell. Okay, this next balance is important, which is the Scara5 or Scara5. It's actually downregulated by green tea or EGCG in green tea. The reason this is important is because of cancer tunneling nanotubes. They're actually upgraded or supported by Scara5. That's why green tea is important to reduce that Scara5. There's also risks associated with down-regulating Scara5 too much, like let's say with excessive green tea. And there are issues if the Scara5 is completely, totally absent. Supporters for Scara5 so that it's not too low, but it can't be too high either, because then the tunneling nanotubes of cancer will start to connect to other cancer cells and transferring mitochondria and PD-L1s to immune cells, attacking immune cells from a distance. Basically, like cancer cells, they actually rescue each other by connecting tunneling nanotubes to other cancer cells by, let's say, transferring mitochondria that they stole from other immune cells. So basically, some supportive nutrients is magnesium to support Scara5, stabilizes the vesicle trafficking. Zinc supports lysosomal cathepsin, and then iron homeostasis. Maintaining iron balance is necessary, but extreme iron overload or deficiency may feed back to suppress Scara5. Now it's careful. You have to be careful to not make the zinc too excessive, because then that's possibly going to increase. What too much zinc is going to end up doing is that it could end up closing chromatin. Your immunity will be weakened, because chromatin has to be opened for gene transcription factors for the immune system to be able to function. And if the immune system is weak, then the cancer is going to thrive, because there's nothing to oppose cancer. Magnesium is also needed to reduce the iron trapped in macrophages. Okay, so magnesium is needed for this thing called FPN1. So basically, if FPN1 trafficking to the surface is blocked, vesicles get shunted to lysosomes for degradation, or the FPN1 that's actually needed on the surface of the cell. So FPN1 is short for ferroportin, and it's the main iron exporter. So FPN1 vesicles are transported along microtubules of actin to the plasma membrane. Therefore, iron is exported from the cell. The consequences of FPN1 loss is iron export decreases, intracellular iron accumulates, or the iron inside the macrophage. Then labile iron pool rises, which means a more oxidative stress, and then the macrophages become iron hoarding and pro-oxidative, or in this case, iron trapped in macrophages. Tumors can actually they can access this iron even if the iron is locked up. So in theory, tumors do this with tunneling nanotubes. So they connect to the macrophage that has the iron trapped, and then that's how the tumor gets the iron from the macrophage in order to support tumor growth. So basically, FPN1 vesicles, which exports the iron, require intact cytoskeletal tract. So low magnesium will actually lead to a low cytoskeletal tracts, therefore leading to loss of FPN1, and that's not good because the FPN1 is needed to export the iron out of the macrophage, so that the macrophage isn't hoarding the iron. Okay, now the effect of green tea EGCG on Scara5 is only effective if there is enough or adequate magnesium. So when EGCG is against Scara5, the transcription of Scara5 is reduced via NFKB suppression and epigenetic tightening of promoter accessibility. Also disrupts lipid-wrapped clustering, which is a scavenger receptors fail to oligomerize. It inhibits ferritin endocytosis without compensatory TFR1 upregulation. Now, some food sources here, besides green tea, since green tea can be problematic with magnesium, if you are deficient in magnesium, is let's say a celery, parsley, and chamomile, because they have luteolin. So they suppress scavenger receptor gene programs, such as for Scara5. It also inhibits RAC1 signaling, which I have videos on RAC1. So basically, luteolin from celery, parsley, and chamomile blocks actin-dependent uptake. So basically, actin is something that actually supports the formation of cancer tunneling nanotubes. So foods like celery, parsley, chamomile are especially valuable when TNTs and RAC1 signaling are consumed. Next one is apigenin, which is are also in parsley and chamomile. They downregulate Scara5 by reducing STAT3 inhibition, or indirectly lowering Scara5 by inhibition of STAT3. Also, another food that can lower STAT3 is also papaya. Papaya does two things at the same time. It lowers STAT3, weakening the Scara5 that the cancer tumor nanotubes thrive on, and papaya also raises STAT5, which improves immune function. Another good one here is resveratrol, which could be found maybe in small amounts in grapes, but it reduces HIF1 alpha, which is a hypoxia marker or induced scavenger receptor expression, reduces ferritin secretion, which means that instead of the iron being secreted, which it's not supposed to do that because it's secreting and exporting are two different things when it comes to iron trapped in macrophage. Resveratrol also improves macrophage ferroportin expression, or the FPN1 that exports the iron outside of the macrophage. So resveratrol and magnesium combined together could be a powerful combo to help export iron outside of the macrophage. Another one is quercetin, because quercetin can reduce high zinc levels. As a result of high zinc levels being reduced, that opens up high zinc, excessive zinc can close chromatin. If chromatin is closed, then the lysosomes, they can't process the iron the way that they're supposed to. So that this iron trapped in macrophage scenario won't happen. So quercetin also chelates iron inside tumor cells, reducing benefit of any ferritin that gets in. So that's it for this video. Like, comment, subscribe, leave your insights down in the comments, and I'll see you in the next video.